Hi class. From Unit 3, you learned how the cardiovascular system can maintain blood volume and blood pressure on a moment-to-moment balance. Now we're going to talk about salt and water balance, which really means long-term balance for blood volume and hence blood pressure. And when we see long-term salt and water balance, we really start to think about the endocrine system and how that influences how the body retains salts, and hence water will follow those salts. And we get a long-term regulation of blood pressure and body fluid volume, and how this can affect the concentration and composition of our body fluid compartments. And I urge you to go back in time and review our body fluid compartments, extracellular and intracellular, and the three compartments for the extracellular. In this picture on the first slide, you're seeing a balance between water input and output. Water input, how do we get water into our blood volume? We get that from the beverages that we drink primarily, secondarily from the foods that we drink, or I should say eat. There's water in the foods that we eat. And then the last part is from metabolism. I'll have you remember that oxygen is the final electron acceptor, and when it accepts electrons and hydrogen ions, it becomes water. How we lose water, primarily that's through the urine that we produce, and then we produce loss of water through insensible losses. That means right now there's evaporation of water through your cutaneous and mucosal linings. That would mean your skin membranes and the mucosal membranes that line, say, your mouth and naso cavities and your lung cavities. You just have water loss. That's different than sweat. Sweat is water loss through sweat glands, and that's very different than water loss through evaporative heat loss through your cutaneous and mucosal membranes, which we call insensible. Even if you're sweating or not, those still happen. And then lastly, you have water loss through your feces. I'm going to cover a lot about the hormones that regulate your blood volume and blood pressure. This is going to be redundant. I'm going to go through this over and over and over again. I'm going to go through each hormone as listed on this screen. But then I'm going to go over how your urine will be changed based on those hormones. So it's really going to be redundant. I'm going to cover how the hormones change your blood versus how the same hormones change your urine. How can you make a more concentrated fluid? We've done this in class many, many times. We have two options, don't we? We can either change the solute concentration or the amount of solvent. If we want to make a solution more concentrated, we can add more solute. And that means we're adding more salt, more glucose. It depends on what we're talking about. If we remove solvent, if we remove water, this can also make a solution more concentrated. But when we do that, we're drastically changing the volume. Conversely, we can make a solution more dilute, can't we? We can get out our molecular tweezers and we can remove the solute, or we can add more solvent. We can add more water. We can dilute it. But if we add more water, this is going to drastically change our volume again. Now you should pay attention to those two last options. If we're changing anything with solvent, this is going to change volume. I'm going to tell you about 4 different pathways that change effective circulating volume, or ECV. The 4 pathways that can regulate how much blood plasma you have, and that's your effective circulating volume, are shown here in the 4 bullets. And you'll notice that 3 out of the 4 are color coordinated in red. The three that are listed first help increase your effective circulating volume. And they all instigate when your effective circulating volume or blood plasma volume drops. So if your blood plasma volume drops, then your blood pressure will drop, and that means your filtration pressure will also drop. And these three first systems will help restore that back up towards normal. The first one is AVP-ADH, and AVP stands for arginine vasopressin, and ADH stands for antidiuretic hormone. The next one is RAA, and we've learned this before. That stands for renin, angiotensin 2, and aldosterone. And I've told you that actually bullet 1 and bullet 2 should go together. It should really be the RAAA mechanism for renin leading to angiotensin 2, leading to aldosterone, leading to ADH. And then the last bullet is the sympathetic output. Again, these first three red bullets show you how these systems are going to restore your blood volume and restore your blood pressure and restore your GFR, glomerular filtration rate. Again, these three systems initiate when your blood volume and pressure and effective circulating volume go down. Maybe you have a hemorrhage. You have to bring it back up to par. The one bullet in black, atrial natriuretic peptide, or ANP, is the one system that helps reduce your filtration load. Maybe your blood volume is too high. Maybe your blood volume and pressure is too high. ANP is going to help lower your blood volume and pressure, lower GFR back down towards a normal filtration rate. So let's look at the three parts of regulating ECV, effective circulating volume, back up towards normal GFR. If we look at this, the first part is a decrease in effective circulating volume. That could happen, maybe our patient has dehydrated, maybe they have sweated too much, maybe they have a hemorrhage, who knows, but they have a decreased effective circulating volume. What are our three pathways? We've got sympathetic influence, we've got RAA, and we've got ADH. And in the middle here we see green boxes, blue boxes, and orange boxes. This represents your three mechanisms to restore blood volume and hence blood pressure and restore GFR back up towards normal. First, what will detect decreased blood volume and pressure? We have pressure receptors found in the aortic arch, carotid sinus, but that's not the only place. We find them in the atria, as shown in the middle blue boxes. We find pressure receptors in the brain. We find also pressure receptors in the juxtaglomerular cells that are found or are part of the juxtaglomerular apparatus. All of these areas are pressure receptors, even in the atria, the cardiac atria. If blood pressure drops because of, say in this example, a blood volume drop, the following changes are going to be instigated. The juxtaglomerular cells, shown in green, are going to detect less volume and pressure. They release more renin. This will lead to angiotensinogen being cut to angiotensin 1. Angiotensin converting enzyme will cut that to angiotensin 2. Angiotensin 2 is a powerful vasoconstrictor. You've heard me say that. That will help change the hemodynamics in the systemic circuit. We're going to get more pressure. We're going to get more vasoconstriction. That means more pressure on the blood that remains in the blood vessels. But this angiotensin 2 can also lead to more aldosterone release, and aldosterone targets the nephrons to increase more sodium and water reabsorption. That's going to help with volume. We also see in the blue boxes that if we have a decrease in blood pressure detected, say in the aortic arch and carotid sinus, we've learned that this will instigate baroreceptor response. This will lead to more sympathetic outflow. More sympathetic outflow means more vasoconstriction. And we also know that more sympathetic outflow will lead to more ADH release, and ADH targets the medullary ducts and makes them more permeable to water. More water reabsorption leads to more blood volume, leads to more pressure. And lastly, if the atria are detecting less venous return because we have a hemorrhage, then the last thing they want to do is release ANP, because ANP will lead to more blood volume loss, and I'll show that to you later. So in blue and green, we're seeing the three mechanisms to lead to more volume, blood volume retention, and blood pressure. And in orange, we're seeing inhibitory outputs because ANP would lead to further loss of blood volume and pressure. And if the instigating stimulus is a decrease in blood volume and pressure, then that's the last thing we want to augment. ADH is made in the hypothalamus, and it's released from the posterior pituitary. ADH, when it's released, causes the medullary duct and collecting duct to insert aquaporins, and the aquaporins will cause the medullary duct and collecting duct cells to insert aquaporins, and this allows water to move from the filtrate through the cells and into the interstitial space. The water is pulled by particles that are accumulating in the inner medulla, shown in this picture on the bottom right. Again, water moves for two reasons, either hydrostatic pressure or osmotic pressure. And the nephron and the collecting ducts and medullary ducts are not cells that are lined with smooth muscles. So there's no hydrostatic pressure to squeeze water out of the filtrate into the interstitial space. Instead, water has to be pulled there, and it's the accumulation of urea and sodium that pulls water there if and when water is permeable. Now, the last thing you want to have happen when you need more water retention is for the interstitial salt gradient to be washed out by the very water you're extracting out of the filtrate. So you still need sodium or urea, you still need some salt to stay in the inner medullary space. At the very same time that ADH causes more water reabsorption through inserting aquaporins, it also causes insertion of urea transporters. And this allows more urea to move from the filtrate out to the interstitial space, which acts as a particle to further extract more water. Again, the medullary duct / collecting ducts are usually impermeable to water. At this point, if there's no ADH left, we have about 15% of our GFR remaining. I'll remind you that in the proximal convoluted tubule, we reabsorb 65% of our filtered plasma water. Then in the descending limb, we reabsorb another 20%. That brings us to 85%. In the thick ascending limb and early distal convoluted tubule, there is no water reabsorption. That means by the time we get to the medullary duct and the collecting, well, I should say the collecting duct, then the medullary duct, we still have 15% of our filtered water remaining. We can reclaim 10% of our filtered water that remains, and that means 5% of our filtered water must go with a waste. That means obligatory water loss. But that last 10% that we could reclaim of our original is only if ADH is present. The collecting duct and medullary ducts, as shown in this picture, are typically impermeable to water. And if we don't have ADH release, then we are going to produce copious amounts of urine that is very dilute. If and when ADH is present, we get the insertion of aquaporins as shown now on the slide. We get aquaporins present on the apical side and the basolateral sides. And this means we can now reabsorb 10% of what we originally filtered in the Bowman's capsule region of our remaining 15%. And if we have those aquaporins present, then we are going to produce a urine that is highly concentrated and scant, very low in volume. And again, as I said before, when we're reabsorbing our water, as shown in the right side of this picture, the last thing we want to do is dilute the very salt gradient that helps pull the water out of the collecting ducts and medullary ducts. We need salts to go along with it. Otherwise, the very pulling power we have will be rendered useless. So ADH or AVP, same thing, causes the insertion of not only aquaporins but also urea transporters. And this means more urea will be accumulated in the interstitial space, and this will further enhance more water reabsorption. The vasa recta, that stands for straight vessels. These are called your countercurrent exchanger vessels. These vessels are found along the very long juxtamedullary nephrons, the very nephrons that we have very few of. These vasa recta will accumulate or collect, I should say, the remaining salt and water that the collecting ducts and medullary ducts retain or have in the interstitial space when ADH is present. So they only account, as far as renal plasma flow, there's only about 5 to 10% of total renal plasma flow that goes through these vessels. Again, most of our nephrons are cortical nephrons, not juxtamedullary nephrons. And as such, only few of our blood vessels going through the kidney are vasa recta. So they're going to only pick up trace amounts of salt and water. The stimuli for ADH are the following. Number one, increased blood osmolarity. If you've got increased blood osmolarity, maybe because you're dehydrated, you've got low blood volume to begin with, and the salts are concentrated in a smaller volume, that's going to drive you to drink water. It might be that you're an uncontrolled diabetic. If you have decreased blood volume, say from dehydration, you're going to have decreased blood pressure. Any of those is going to drive you to seek water. But there are other stimuli. There's dryness of the mouth. Maybe you're nervous, you have to give a speech. That will drive you to seek out water. Maybe you're sick and feverish. That will seek you to drive out water. Burns is going to lead to blood volume loss. Excessive sweating, I've already mentioned that. Vomiting, diarrhea, anything that can lead to volume loss will lead to increased ADH secretion. And that means not only will your nephrons retain more water, but you will seek out more water to drink. Conversely, things that decrease ADH and will in turn decrease thirst. If you have increased blood volume, maybe you've consumed too much water and you have too much blood volume and you have too much blood pressure, and from all this excessive water that you consumed, you have decreased blood osmolarity. All of those things are going to decrease ADH. The last thing you want to do is retain more water and seek out more water. Strike that out. Other factors that are limitations to ADH release are alcohol. Alcohol inhibits ADH release, and if you inhibit ADH, that means the collecting ducts and medullary ducts are not permeable to water, and that means more water loss in the plasma or in the urine. If you've eaten too much and you have gastric distension, that will inhibit ADH. Again, the last thing you want to do is retain more blood volume when you feel overly full. A decrease in ADH means copious amounts of dilute urine. In our next segment, I'm going to continue with the next bullet that leads to more blood volume and pressure, and that's the RAA pathway, renin-angiotensin-aldosterone pathway.